This is my area of interest is precision pharmacology, and we'll also discuss the next gen therapeutics that are currently being employed in the field of veterinary medicine and veterinary pharmacology. For starters, I want to introduce you to what is precision pharmacology. As you have heard in the series of VetExcel, so we are discussing about advances in pharmacotherapeutics or advances in veterinary sciences. So precision pharmacology is basically a branch of precision medicine. In veterinary science, we are dealing with precision veterinary pharmacology. I want to imagine yourselves as buying some clothes. Like if you want to purchase some clothes, you go to the market, you have ready-made clothes. Okay, so your ready-made clothes are like different categories into sizes like small, medium, XL.
Hello, sir.
Hello, sir. Hello, sir. Hello, sir.
Hello.
Hello.
हाँ सर.
Was there an interruption? कुछ interrupt हुआ क्या?
No, I
Hello.
think you are exit from the meeting.
Okay, okay. Where were we discussing? Like,
Your sir, your screen not visual, sir.
okay, just a second, just a second. My apologies.
हाँ sir.
Yeah, is it visible now?
हाँ sir, now visual, sir.
So actually what we were discussing was like the difference between conventional and precision pharmacology. Like I was saying an example of buying clothes. Like if you go into a ready-made store and get yourself a set of clothes, like you choose based on your general size. Like there is some measurement for small, medium, XL, double XL, some measurements, industrial standards. So you buy those clothes that fit you. There may be some fitting issues, some may be oversized, some may be there is some issue with the proper size of clothes. So you can just think conventional pharmacology as a ready-made clothes, whereas precision pharmacology is a tailor-made clothes. Like you go to a tailor, get your measurements done, stitch your fabric, choose the kind of fabric that can suit you, and get it done according to your convenience, like how lengthy you want to keep it. So it is a tailor-made medicine. That is what precision pharmacology implies. Like this is the basic difference between a conventional pharmacology and precision pharmacology. In medicine also, like in conventional pharmacology, we give the drug to the animal based on body weight or body surface area. Like most of the medicine you take is based on body weight criteria. Like suppose for an adult, this is the body weight, and your doctor will give you dose accordingly. Whereas in precision pharmacology, we choose the right medicine, like if the medicine is suited to the animal, right animal, like which species, which breed, which age, everything is calculated. So this is a tailor-made medicine, you can say. Coming to the definition, the precision veterinary pharmacology, it refers to a customizing drug therapy based on specific genetic makeup or metabolic pathway of an animal or disease susceptibility of that particular animal. So it is a tailor-made medicine applicable to that particular animal. That is what precision medicine or precision pharmacology stands for. What challenges do we face in conventional pharmacology, which necessitates the need of a precision medicine? Like when we have conventional pharmacology, which is currently being followed everywhere, why do we need precision medicine? That is the question. So there are some disadvantages with conventional medicine. For suppose starting with an empirical dosing, like I said, conventional medicine is my apologies for the internet dysfunction. There seems to be an issue with connectivity. I'll try my best to present the lecture with minimum interruptions. So, if you can confirm whether my slides are visible.
हाँ sir, visual sir.
Thank you very much. So, as I was telling you, what are the challenges with the conventional approach? Like when we are having a conventional setup, conventional traditional pharmacology approach, why do we need to go for a precision medicine? So as I was telling you, the conventional pharmacology, it employs a one size fits all approach. For example, just I stated, the ready-made clothes, like they are manufactured in a large scale in industry. They do not consider one particular individual. They manufacture it for a whole community. So that is what is here reliance on average dose ranges without considering individual variability. Like they do not consider individual variations. The main criteria employed in traditional veterinary approach is you dose the animal based on body weight. Like what is the average body weight of an animal, and depending on it, you dose the animal. Whereas in precision pharmacology, you get to customize what is the type of individual, because even in same body weight individual, there may be variations. They may have different enzymatic pathways. The metabolizing enzymes may be different. The target cell target may have some variations, molecular variations. That is why we are suggesting a precision veterinary dosage is superior to empirical dosing. Second thing is resistance development, especially in case of microbes, where the development of resistance is common nowadays because of broad spectrum drugs and the general usage without taking into consideration the individual variations in the microbe. Complex and chronic diseases such as autoimmune disorders, osteoarthritis, neoplasia, or some metabolic syndromes. These diseases are individual specific because some genetic variation in the individual or molecular variations. So these diseases, they do require the specific approach rather than a general approach. I'll also discuss the examples regarding complex chronic diseases in my upcoming slides. And the next challenges with conventional therapy is frequent reporting of adverse drug reactions. Because in conventional drug therapies, the individual variations to drugs are not considered. So there is a chance of increased concentration, plasma concentration. Thereby, the animals may experience toxicity, or sometimes the subtherapeutic dosage may be reported in the animal, which leads to the failure of therapy. So therefore, uh, experiencing toxicity or receiving subtherapeutic doses all lead to failure of therapy. That is what commonly reported in conventional pharmacology. And conventional pharmacology also integrates poorly the omics technologies like genomics, proteomics, metabolic data, metabolomics. Everything is taken into consideration, and it's not in conventional pharmacology. This takes us to the need for so as we following this, the need for because there is a diversity that you cannot even exhibit genetic flexibility, which can influence the drug metabolism, efficacy, and the course of treatment. Like, suppose for example, there is a variation of ABCB1 gene, like it is the antibody, the ATP binding cassettes B1 gene mutation in suppose herding species like coolies or Australian shepherds. So there is a variation in ABCB1 gene. So this caters to a protein called P-glycoprotein, which is involved in drug transport. So this P-glycoprotein is an efflux pump. So the drug transport across blood-brain barrier is affected in these species. Okay, so that may lead to toxicity. Even at normal doses, if you compare it with the other animals, the same body weight, if you give that drug in breeds such as coolies, it may cause toxicity. And also some variation in gene like cytochrome P, CPY2D6, which is responsible for metabolizing of the drugs. So there is a alteration in drug metabolism because of which there is an increased plasma concentration reported. So even if there is a genetic difference in metabolizing enzymes, there may be increased plasma concentration or altered drug metabolism leading to toxicity. So that is the genetic diversity, which is taken into consideration in precision pharmacology. Adverse drug reactions, like I said, there are reports of adverse drug reaction with the conventional pharmacology. Like in precision pharmacology, we can identify the high risk individuals, like screening for genetic markers, like the MDR1 gene mutation. We can screen the animal and we can predict the toxicity causing factors in such animals. Moving to antimicrobial resistance, like I said, in conventional pharmacology, there is a risk of increased resistance. Whereas in precision medicine, we are having a targeted therapy that minimizes the selective pressures, like we are not allowing resistant organisms to survive. So we are removing the selective pressure for bacteria to survive and multiply, thus preserving the efficacy of already existing drugs. Okay, we can make nanoformulation, we can deliver them to the target microorganism, improved efficacy. So all this can counter the emerging resistance. Also, economic impact of precision pharmacology is also there. Like precision approaches, we can reduce the chances of treatment failure. So therefore, we can improve the productivity in livestock and food industries. We can save the cost of drug treatment. We can also minimize the withdrawal time in case of milk or meat. Thereby, we can help the farmer or livestock owner economically. That is why there is an economic impact of precision medicine. And from the animal welfare perspective, tailor-made therapies can enhance the recovery rates and minimize suffering. Like I have examples of some tailor-made therapies that can enhance the recovery in regenerative medicine. Like precision medicine also supports the ethical stewardship or ethical use of pharmaceuticals, aligning with the welfare standards of global organizations. So taking into consideration all of these points, there is a need for development of veterinary precision pharmacology. So, what are the methods that are commonly employed for precision pharmacology? We have discussed here, like the pharmacogenomics approach, nanomedicine, stem cell therapy, gene editing, molecular profiling, AI diagnostics driven precision medicine. All of these come under the next gen therapeutics that are contributing towards the precision medicine. Like the speaker just before me was telling about nanomedicine, which is also a part of precision medicine. So in general, we'll look into each of these approaches. First, starting with the pharmacogenomic approach. Pharmacogenomics is considered to be a core pillar of precision pharmacology. In pharmacogenomics, we deal with the genetically influenced variation to a drug response. Like we study how genetic variation in an individual can influence the response of that individual to the drug. Responses including the absorption patterns, distribution, metabolism, or excretion patterns like the pharmacokinetic variability or the pharmacodynamic response of an individual to a drug that is determined by its genetic makeup. In veterinary medicine, particularly, as we are dealing with multiple species like canines, felines, equines, bovines, all the other species. So there is a varied range of animals that we are dealing with. So the genetic makeup also varies accordingly. And even within a same species, there is a breed specific variations that can affect your treatment. And within an individual, like same breed, there is metabolic pathway may differ that can affect your treatment protocol. So all these come under the pharmacogenomic studies. So pharmacogenomics, it helps us in predicting the toxicity or treatment protocol. Like we can understand the polymorphism in genetics. Like example, well-established polymorphism is of MDR1 gene and also the cytochrome gene, CYP genes. So by determining these variations, we can identify the animals that are at risk of drug toxicity, and therefore we can optimize the choice of drugs. Suppose if an animal is poor metabolizing agent of is a poor metabolizer for certain drug, we can choose the alternative drugs. So based on molecular markers, we can employ the targeted therapy, and based on genetic makeup of an individual, we can have individual dosage adjustment. We can design targeted biological agents like as the monoclonal antibodies. So all these we need the pharmacogenomic data. For example, like I said, major pharmacogenomic approaches that are currently being employed in the veterinary medicine includes the MDR1 gene. MDR1 gene is a well-established marker that we are looking for as a drug transporter. So the gene is ATP binding cassette B1. Formally, it is known as MDR1, that is multi-drug resistance 1 gene, and the protein that it codes for is P-glycoprotein. So it is basically a drug efflux transporter in species such as dogs, especially the herding breeds like as a collies, Australian shepherds, sheep dogs, etc. There is a variation of ABCB1 gene. So the drugs such as ivermectin, doramectin, moxidectin, and the loperamide, vincristine, doxorubicin, all these drug metabolisms are influenced by the P-glycoprotein in the animals. For example, the ivermectin, if you determine the dosage of ivermectin, 200 micrograms per kg or somewhere around that. If you give the normal dosage in Australian shepherds or border collies, etc., you can cause toxicity in those animals because the efflux protein that is responsible for pumping out of the drug out of CNS is deficient in them. So even at low doses, you can incur the toxicity in these animals. So before dosing, you have to understand the molecular mechanism, the genetic makeup of the individual, then you have to go for dosing. Second most common mechanism or pharmacogenomic principle involved is CYP450, the cytochrome P450 polymorphism that can alter the drug metabolism. Genes like CYP2B11, 2D15, 3A2, all these are coded for drug metabolizing enzymes. In species such as dogs, they can influence the drug metabolism of certain drugs that are commonly encountered like phenobarbitals, your NSAIDs, your ketoconazoles, or your anesthetic agents. Based on the polymorphism, the animals can be classified as slow metabolizers or fast metabolizers. Like in slow metabolizers, what happens is the expression of these genes is decreased, thereby the enzymes that are responsible for drug metabolism also decrease. So in case of decreased drug metabolism, the concentration of drug remains high in plasma. So even at low doses, there is a risk of toxicity. Like if you go for conventional approach based on body weight, and you inject the animal or give the animal certain dosage of drug, if the animal happens to be a slow metabolizer, there is a risk of toxicity because the drug is metabolized slowly and the plasma concentration is relatively high. Whereas if the animal has increased expression of the cytochrome enzymes, it is called as fast metabolizer. In fast metabolizers, the concentration of enzymes is high. When you go with the standard dose calculated with the conventional pharmacology, there is a risk of subtherapeutic dosing. That means your plasma therapeutic concentration will never reach the optimum levels. Therefore, the therapeutic plasma concentration is very poor and it will not even cause an effect. Therefore, it may result in the therapeutic failure. So these effects has to be and taken into consideration. So the genetic variation, unless you take the genetic variation into consideration, your treatment will not succeed. Second is a kit, proto-oncogene c-kit. It is in cancer medicine. We target this gene. It is a tyrosine kinase receptor linked gene. Mainly, we can see this gene in canine mast cell tumors. So we also have one drug that is recently approved by the FDA. Tosiranab is the drug. It is with the brand name Palladia. So it is the FDA approved veterinary cancer drug which targets the c-kit mutation in case of canine mast cell tumors. So in oncology also we are targeting the modifications of the tyrosine kinase gene and the specific drugs we are synthesizing. This is counters for precision molecular oncology, taking into consideration the pharmacogenomics of an individual. Next, the interleukin 31 pathway. It is a target for canine atopic dermatitis. As we can see, most of the cases of canine atopic dermatitis clinically is mostly managed by conventional therapy using steroids. Okay, the steroids, they may cause the mask the symptom, but the threat of interleukins is always there. So what they do is in precision medicine, they are targeting canine specific monoclonal antibodies that can neutralize the interleukin 31. Therefore, they are decreasing the inflammatory mediators. So it has a minimal systemic immunosuppression. In case of steroids, as we use it without discretion, so there is a systemic immunosuppression. If you give animal steroid, there is overall immunosuppression in the body. Instead of that, we can go for interleukin 31 blocking. That is a precision medicine. So it also has a longer duration of action compared to a conventional therapy. Systemic use of steroids also has a toxic side effects that can be minimized using the interleukin 31 pathway targeting. So specifically targeting canine immunopathways, you have a better medicine and a precise medicine compared to the conventional therapies. Additionally, there are also some emerging pharmacogenomic targets that are being currently researched for various drug metabolisms, like cyclooxygenase polymorphism in dogs is being evaluated for NSAID therapy, that is in pain management, and experimental monoclonal antibodies targeting HER2 for canine mammary tumors, and EGFR tyrosine kinase inhibitors for oncology, and BRAF mutation for transitional cell carcinomas. These are being explored as a targeted therapies for various disease diagnostics and disease management. This all are possible due to the knowledge of pharmacogenomics. Next thing is, next area of precision medicine is nanomedicine. Like you might have listened to the speaker that spoke before me about nanomedicine, particularly. So nanomedicine or nanotechnology, up to 1 to 100 nanometer scale medicine is used in the diagnosis, drug delivery, and therapeutics. So it has improved the targeted drug availability, increased the bioavailability, promoted the sustained release products, thereby reducing the toxicity and improving the vaccine delivery, or all these areas that are positive effects of nanomedicine. Some key applications we are discussing is tumor targeting. There is one preparation of liposomal doxorubicin that can target the tumor cells like canine lymphomas and hemangiosarcomas. So there is a targeted drug delivery to tumor cells. Increased drug delivery and efficacy of treatment is there. We can also have nanoitraconazole compared to a conventional itraconazole. Nanoitraconazole has improved oral bioavailability. Therefore, the dose of itraconazole required is also low. And the antifungal drugs have notorious for hepatotoxicity. We can reduce this hepatotoxicity risk using the nanoitraconazole. So it is applied mostly in dermatophytosis in dogs and cats. So this is a conventional alternative to conventional therapy. We have silver nanoparticles. Most of the clinical sprays, topicals application nowadays contain the silver nanoparticles. The main target of the silver nanoparticles is the bacterial cell wall and membranes. It is also like promoted that these silver nanoparticles can disrupt the biofilms produced by the bacteria as a protective layers. So in post-surgical wounds or in mastitis, you can nowadays see the silver nanoparticles, which is a nanomedicine based product. Next area of research is the nanovaccines in livestock. These nanovaccines particularly target the antigen presenting cells, and conventionally we are using chitosan derived nanoparticles for antigen delivery to these antigen presenting cells. This is currently employed in foot and mouth disease. Some nanogels are also being invented, like for use in mucosal membranes of the animals. Poultry viral infections, bovine respiratory disease complexes are all targeted by these nanovaccines. Next therapy of precision medicine is the stem cell therapy. Stem cell therapy is particularly of important in regenerative medicine. So it contributes to the precisive and regenerative medicine by its application in tissue regeneration, the usage for immunomodulation, and the anti-inflammatory effect. It also decreases the long-term drug dependence of the individuals. The one common area that is employed is the osteoarthritis treatment. We have the articular cartilage degenerative diseases, synovial inflammation, etc. are being treated by these stem cells. These stem cells, again, we have mesenchymal stem cells, adult stem cells that are derived from adipose layers, etc. Or there is also investigation of embryonic stem cell usage. The approved products include, one of the products is RT-cell4, approved by the European Union for equine lameness due to osteoarthritis. We can also employ stem cells for tendon and ligament injuries, particularly in sports medicine, sports animal medicines. We are using this superficial digital flexor tendon injury, suspensory ligament injury. There is autologous bone marrow derived stem cells, or autologous, that is from the same animal. We can derive the bone marrow, isolate the stem cells, and inject back into the animal. So that is a mesenchymal stem cell injection. We can also go for autologous adipose derived stem cell injections also. That is the mesenchymal stem cell therapy. Embryonic stem cells are currently under investigation because embryonic stem cells have tendency to become oncogenic. That is, they can transform into cancer cells. That is why adult cells are being preferred over embryonic stem cells. Certain products like the RT-cell4s in horses, or autologous mesenchymal stem cells, allogenic mesenchymal stem cells. Allogenic is from the different animal. So all these are being investigative in investigative stages for treatment of osteoarthritis or immune complex mediated diseases. We are currently investigating the stem cells. Another approach is exosome based therapy. Here what we are doing is, when we are going for stem cell therapy, there is a certain complication that is associated with delivery of cells. Like you are delivering a foreign cell into the body. So there is immunomodulatory reactions that can happen, or the stem cell can transform into cancer cell, which can affect your therapy. So you are injecting a cell for regenerative therapy, and that cell can become oncogenic and it can cause cancer. So to negate all those complications, we can go for the exosome based therapy, where there is a cell free approach. We can take the cell secretome, that is what we call the contents that are secreted by these stem cells. So we can take that exosomes, that is a vesicle that contains the secretome. So exosomes or secretome based cell free approach. Here we eliminate the foreign cells. We only have the cell secretions that can target the inflammatory pathways or tissue repair mechanisms. And instead of administering the whole stem cells, the purified vesicles derived from stem cells, if we are using it, we can have the lower risk of immunogenicity and tumorigenesis risk. They are also easy for storage compared to the live cells, because for live cells you have to store them, you have to revive them back to their original condition, etc. There are complications associated with the stem cells. So if you are using cell derivatives, there is a reduced risk of immunogenicity. And the other area that is contributing to the precision medicine is the gene editing. Gene editing, it refers to the modification of DNA with the genome tools in order to, it can be employed to correct mutations, or it can be used to knock out any harmful genes, or for insertion of beneficial genes, to regulate what area of DNA is to be expressed. So after the popularization of CRISPR-Cas9 technology, this gene editing has become a major tool in achieving precision medicine. One certain area that has worked regarding the gene editing is PRRS virus, that is the porcine respiratory reproductive syndrome virus. So here we are targeting the CD163 gene. So porcine reproductive and respiratory syndrome disease, we are what we are doing is, it is using the CRISPR-Cas9 technology. We are deleting the viral binding domain, that is where the viral binds and causes the disease. So in PRRS resistant pigs, the viral binding domain is absent, so the pigs are resistant to the infection. So therefore, we are reducing the economic losses associated with the disease, and also we are reducing the antiviral drug usage, because when the virus infection is not there, there is no need to use antiviral drug usage, or we can also minimize the public health risk by using this gene editing technology. Another area is BRAF mutation. Like in pharmacogenomics, I've told you that BRAF contributes to the transitional cell carcinoma. So BRAF mutation we are targeting, we can reduce the transitional cell carcinoma occurrence. Gene editing correction by tyrosine kinase inhibition gene we are targeting there. So it is still under investigative stage. So BRF mutation in canine bladder cancer is also an area for gene editing technology to be employed. Now, as we have discussed all the positive points regarding the precision medicine, so if there is so much positivity with the technology, has it been limited? Question is, the major limitation of high is for this employment of this precision medicine is the high cost. Like in countries such as ours, where the average income of the farmer is not that much, so the cost of technology is the major contribution of it application. So this precision medicines, like I discussed, genomic sequencing, biomarker testing, stem cell isolation, stem cell expansion, all these require sophisticated laboratories, like the nanomedicine production. All these are technology intensive domains. We have to invest heavily in the infrastructure first to make this economical. Currently, we do not have the required infrastructure for large scale expansion of these technologies. We do not have genomic sequencing facilities. Forget about animals. In humans also, we do not have proper genomic sequencing facilities. Stem cell technology is currently in investigative stages in the human medicine also. I think only approval is for hemopoietic stem cell transpositions they are approving. For mesenchymal stem cells or embryonic stem cells in human medicine, there is currently other concerns also, like I'll discuss in ethical concerns. So these are all some high intensive technologies that require capital expenditure. So these are currently limited by their cost. Other major limitation is the availability of data. Suppose if you are not having whole genome sequencing facility, that is very expensive. So all these species specific data you cannot have, like the genomic and biomarker data across species. And also we do not have proper databases constructed for species wise variations, and our validation studies are insufficient. All these can limit the predictive ability or accuracy of your study, and therefore you do not have a reliable data for application of the precision medicine. And in veterinary medicine particularly, there is a lack of standard protocols, particularly in stem cell medicine also. There is lack of protocols regarding how much dosage is to be employed, which route of administration is sufficient, uh, what is the treatment frequency, like how many doses are required. All these data are lacking. So the results produced by one lab may not be reproducible in the other labs. So we do not have proper standardized protocols established by the regulatory agencies. And also we have legal and ethical concerns like gene editing, stem cell therapy. There are uh, concerns regarding the ethical use of stem cells, or there is unclear approval, like how to, if you are inventing a stem cell therapy, like how to apply for its licensing, which category to apply, biologicals or conventional medicines. And there is differential regulating authorities also there, which to which authority you have to apply. And all the ethical concerns are associated with embryonic stem cells is also there. If you are like using embryonic stem cells, how you are sourcing the embryos, that is also a major concern that limits the certain therapies. Like suppose gene editing livestock, many people are not favor for gene editing. In plants also gene editing is employed. There is ethical concern regarding gene edited varieties of plants, and also public skepticism towards genetically modified animals. So this can like contribute to the negative side effects of, like negative limitations of the precision medicines. In conclusion, I can say that precision medicine is a futuristic concept. It is currently in research stages. We hope that the precision medicine, even though it is beneficial from the point of a veterinary doctor, or from the it is also like cost effective from a livestock owner perspective. Currently we do not have all the requirements to fulfill for employment of these precision medicine. So in healthcare strategies we have to include capital expenditure, is promote the expenses, or promote the technologies that can um, make these precision medicine available to the animal. So for widespread implementation, it is currently constrained by high cost and all the limitations that we have discussed so far. But in future, this technology, I hope so, it is implemented for the welfare. Thank you, everyone. If you have any questions.
Thank you, sir. Anyone? If you have any query, you can ask.
If you can come up with any question, you can just, I'll ask the moderators to share my email. You can just email me. I can provide you with the relevant data articles regarding the same.
Sure, sir. Anyone have any query? You can ask now.
So this precision medicine is available at lab level only, right? We can do for research and other purpose, but practically at field level, for us, it's not available, right?
Some of the technologies are at lab level, but certain therapies in advanced countries like Europe, they are employing the stem cell therapies. They have, like I mentioned, RT cell food is a precision medicine technology, but it is available at the field level, because the